Hello, my name is Benedikt. I am a senior at the University of Massachusetts Amherst, and I work in Dr. Madeline Bartlett's lab, which researches grass evolution and development. The title of my research presentation today is Investigating an Agriculturally Important Grass Trait with CRISPR. I will go through this talk in three parts. First, describing the species I work with, Brachypodium distachyon, and the specific organ that I'm interested in, the awn. Second, I will go through awn evolution, function, and a certain gene required for awn development in barley. Third, I will describe my own work attempting to create knockout mutants of the Brachypodium short internodes genes, but for now you don't need to know any of that. What you need to know is I work on a small model grass called Brachypodium distachyon. This species is an ideal model organism because of its small size, small genome size, short generation time, and established CRISPR-Cas9 transformation protocol. In addition, it's closely related to barley, wheat, oats, ryegrass, and many other foraging grasses in the Puidae subfamily, which makes Brachypodium an excellent model organism for answering questions related to these very important grasses. Now, what are awns? Awns are a thin, hair-like extension of some grass seeds, and even between grasses, awns have extremely varied forms and functions, as you can see below. Awns are thought to provide the seed a variety of benefits based on their function, including seed dispersal, establishment, and maturation. For example, this twisted awn on the right actually unwinds in response to moisture and drills the seed into the ground, effectively planting it. Now that I've discussed Brachypodium and awns on their own, I'd like to combine them. Brachypodium individual flowers, or florets, are arranged in spikelets, as you can see on the left. This spikelet contains 2 rows of florets, and each has an awn indicated by these red arrows. As you can see on the right, grass flowers are organized much like other flowers with pistil, stamens, and surrounding organs. However, grass flowers are encapsulated by a lemma and a palea on either side. These structures will eventually become the so-called husk of the seeds, and the awns in Brachypodium extend out from the lemma. Straight awns such as those in Brachypodium distachyon and barley are hypothesized to be involved in seed establishment and potentially photosynthesis, making them an important trait for investigation. So, I just gave you all an overview of Brachypodium as well as a description of its awns. I will now describe some aspects of awn evolution, hypothesized function, and a specific gene required for awn development in barley. So, to start out with awn evolution, below is a grass phylogeny focused on the Puidae, the subtribe containing wheat, barley, and Brachypodium. The circles at each node or intersection represent an ancestral state reconstruction that predicts the likelihood of awns being present or not in recent ancestors based on their modern descendants' awned or awnless states. This is excellent work done by Aaron Patterson in the Bartlett lab, and I have one main takeaway for my project, which is that the closely related barley, wheat, and Brachypodium distachyon share a likely awned common ancestor, which suggests a shared function between the awns in these species. And I'm getting ahead of myself here, but this also suggests a possible homology between the genes that control awn development. That was all on awn evolution. Now I wanted to show some data that is known about the agriculturally important grass species barley and wheat, and what happens when their awns are clipped. These graphs are a little bit difficult to read because they were drawn by hand, but the important takeaway is that by clipping awns off, they reduced yield in both grasses. On the left, the percentage of decrease in yield in wheat was higher the earlier that they clipped awns, and on the right, the weight of barley kernels was lower in clipped spikes compared to unclipped ones. This data suggests that Puidae awns assist in seed maturation and that the same might be true in Brachypodium. Having just described awn evolution and function briefly in the Puidae, I would now like to pivot slightly to describe an interesting barley mutant to you. The LKS2, or short awn 2 mutant in barley, has around 50% shorter awns and altered pistil morphology. That's another floral organ if you remember from earlier. So, the LKS2 gene is clearly involved in floral organ development and specifically in awn elongation in barley. The LKS2 gene is also a short internodes family transcription factor, and transcription factors generally have a higher impact on development than other genes. This begs the question, is there a homologous gene regulating stimulating on elongation in Brachipodium distachyon that we could investigate to determine how conserved the pathway for on development is in the Puidae. I'm glad you asked, because there is a homologous gene, in fact, multiple. Below is a gene tree or phylogeny of short internodes genes, or shy genes, in Brachipodium, barley, and several other plant species. This shows how similar and likely related the short internodes genes are to one another, and which genes between species are likely homologous. There are 4 Brachipodium short internodes, or BD shy genes, 3 of which are more similar to LKS2, our barley gene, highlighted in blue at the top, than the 4th. Therefore, I hypothesized that a Brachipodium knockout mutant with 1 or more non-functional copies of these BD shy genes would likely have a short on phenotype similar to the LKS2 mutant in barley. Up until now, everything has led to my big question, which is, how conserved is the genetic pathway for on development in the Puidae subfamily of grasses? My hypothesis on this question is that the Brachipodium distachyon short internodes genes share a role in on elongation with short on2, or LKS2, in barley. Finally, my experimental approach will be to evaluate the BD shy 1, 2, and 3 mutant phenotypes and compare them to the LKS2 mutant phenotype in barley. At this point, we've reached the end of my outline and will now finally begin the actual work that I did, which included attempting to create knockout mutants of my 3 BD shy genes using CRISPR-Cas9 genome editing. If you're not entirely familiar with CRISPR-Cas9 genome editing, I will give a short description of how it works. CRISPR-Cas9 genome editing happens when a small target gRNA or guide RNA binds to double-stranded genomic DNA and pulls down the CRISPR-Cas9 protein, which then cleaves the DNA to create a double-strand break. As the plant or whatever other organism hastily attempts to repair this break, it often incorrectly repairs its own DNA, leaving a mutation near your target site. Now, since this process relies on the plant's own DNA repair mechanism for the mutation, all that needs to be expressed in a cell is the gRNA and the Cas9 protein itself. Now the question becomes, how do we get this DNA into the plants to be expressed in the first place? Well, in the Bartlett lab, there's a strict protocol including plasmid design and agrobacterium-mediated transformation for Brachipodium genome editing. First, a promoter sequence is combined with a guide RNA that includes a target sequence to bind the plant's DNA. Then, these multiple target guide RNAs are combined into one larger plasmid called p-enter. Then, those promoter and guide RNAs are all moved into a plasmid with the Cas9 protein base. This final plasmid with your target guide RNA and your Cas9 protein base is put into agrobacterium, which will insert all of these DNA sequences into the plant's genome where they will be expressed to mutate your genes of interest. The process I just described was lengthy, but a member of the lab named Aaron Patterson had previously created a final plasmid with 3 spacer sequences to target the 3 Brachipodium distachyon short internodes genes. I performed an agrobacterium transformation with this plasmid, then regenerated and genotyped the 49 resulting plants. Each plant had to undergo genotyping PCR at 3 different locations in the genome, taking several months, and yet there were no mutant alleles recovered. So, with no mutants being generated from the previous plasmid's plant transformation, I set out to create a new CRISPR plasmid to knock out the BD shy genes with new targets and multiple targets per gene this time. In this case, the target sequences were around 200 base pairs apart, ideally creating a knockout in the first exon of each gene that will result in a completely non-functional protein. However, during my time trying to assemble this 6-spacer plasmid, I made progress but was ultimately unsuccessful because of the high number of inserts. I tried several methods to troubleshoot this issue, but after several months I eventually changed my approach. So I pivoted to target fewer short internodes genes, leaving out BD shy 3, which is most closely related to another barley gene, VRS2, and less likely to share function with the other 2 if they are involved in on elongation. After reducing the number of inserts from 6 to 4, I was quickly able to make my final plasmid and have since transformed it into agrobacterium, which I then used to perform a plant transformation. On this slide, I've shown that the ideal plasmid I assembled online on the left matches the sequencing from my real-life plasmid perfectly. After transforming the plant tissue, I have performed several steps of tissue culture, essentially moving tissue from one plate to another, and am currently waiting for plants to regenerate before I can begin genotyping. Once the plants have grown enough for me to take tissue samples, I will be genotyping each plant at the target loci, and hopefully, I will have an interesting new mutant phenotype coming soon that I can use to make inferences about the roles of the BD-shy genes. I'd like to thank all the members of the Bartlett Lab, the University of Massachusetts Amherst, the Center for Agriculture, Food, and the Environment, and the late Ray Ethan Torrey for all of their support of my research project.